Diet and eye pressure, how proteins, fats, and carbs may affect glaucoma. Glaucoma is a leading cause of irreversible vision loss, usually caused by damage to the optic nerve, often driven by high intraocular pressure, IOP, the fluid pressure inside the eye. Lowering IOP is the main way to treat glaucoma, but eye pressure can be influenced by more than just medications. Recent research suggests that what we eat, especially the balance of proteins, fats, and carbohydrates, may play a role in eye pressure and glaucoma health. In particular, certain dietary patterns, for example, low-carb or Mediterranean-type diets, have been linked to glaucoma risk, and measures like nerve fiber thickness and visual field loss. At the same time, scientists have begun to uncover biological pathways, from blood sugar and osmotic pressure to insulin effects and lipid signaling, that could explain how diet affects eye fluids and drainage. This article reviews the latest evidence on macronutrient patterns and glaucoma. We will survey epidemiologic studies of diet patterns, low carbohydrate, low fat, high protein, and Mediterranean-style diets in relation to glaucoma, nerve thickness, the retinal nerve fiber layer, and vision loss. We will also explain possible mechanisms, including osmotic shifts from sugar, insulin's effects on eye fluid, and the role of fats and lipid signals in the eye's drainage mesh, that might link diet to IOP. Finally, we highlight gaps in the research, notably the lack of long-term trials, and suggest ways future studies can standardize diet tracking and glaucoma measures to get clearer answers. Dietary patterns in glaucoma, what the studies show. Low-carbohydrate diets. The idea of a low-carbohydrate diet, shifting calories from carbs to more protein and fat, has been widely studied for weight loss and diabetes, but does it affect glaucoma? A large U.S. study examined over 185,000 adults over decades and tracked their diets and glaucoma outcomes. That study found no overall link between long-term low-carb eating and the risk of primary open-angle glaucoma. In other words, simply eating a low-carb or ketogenic-style diet did not clearly reduce or increase glaucoma risk in most people. However, this same research did find an intriguing hint. If people substituted more vegetable-based fats and proteins, like plant oils or beans, for carbohydrates, they tended to have a lower risk of a specific glaucoma pattern, one that affects central vision early. In practical terms, swapping plants and healthy fats for carbs might modestly protect against one subtype of glaucoma. In contrast, sugary or high glycemic carbohydrates seem to raise eye pressure acutely. For example, one Taiwanese health study measured people's blood sugar 2 hours after a standard meal and compared it to eye pressure. They found that participants with higher post-meal blood glucose levels had significantly higher IOP by several millimeters of mercury than those with lower glucose. Each rising quartile of after-meal sugar gave a clear trend of higher eye pressure. This suggests that spikes in blood sugar, which happen with high-carb meals, can temporarily increase IOP. In fact, classic studies in diabetic patients have shown that acute high blood sugar makes the eye fluid more concentrated, higher osmolality, shifting water and raising IOP. Thus, diets very rich in sugar or refined carbs could raise osmotic pressure in the eye fluid and elevate IOP. By contrast, low-carb diets may avoid those sugar spikes, but long-term data have not consistently shown a protective effect on glaucoma overall. Low-fat diets. Low-fat diets have also been tested in relation to glaucoma. In one large secondary analysis of the Women's Health Initiative trial, over 23,000 women randomized to a low-fat diet versus usual diet, researchers looked at new glaucoma diagnoses for about 12 years. They found no reduction in glaucoma from the low-fat intervention. In fact, the hazard ratio was about 1.04, meaning essentially no difference. Interestingly, further analysis showed that for women who already ate very low fat at baseline, the supervised low-fat diet increased glaucoma risk, about 22% higher. In plain language, cutting fat, especially in people who were already on a low-fat regimen, seemed to backfire. Overall, however, this trial suggests that a broad low-fat dietary pattern, more fruits, veggies, and grains instead of fats, did not meaningfully lower glaucoma incidence. In contrast, some cohort studies of fat intake have suggested subtle links between fat types and glaucoma. For example, one U.S. prospective study of health professionals found that a diet very high in omega 3 fats relative to omega 6 fats was associated with an increased glaucoma risk. This was surprising because omega 3s are often thought healthy, but perhaps the interplay with omega 6 is complex. Another analysis of U.S. nutrition survey data found that higher intake of the fish oil fats EPA and DHA, types of omega 3, was linked to lower odds of glaucoma, while higher total polyunsaturated fat intake, omega 3 plus omega 6, correlated with higher glaucoma risk. These findings are not entirely consistent, but they highlight that fat type matters. Omega 3s from fish oil may help, whereas diets overly rich in certain edited fats might not. High-protein diets. What about protein-heavy diets? Observational evidence is limited. A few cross-sectional studies, mostly in Asians and Europeans, have found that people with glaucoma tend to eat less meat and protein than those without glaucoma. For instance, a Japanese study reported that women who ate meat more days per week had about 40% lower odds of open-angle glaucoma. Similarly, researchers in Greece noticed that glaucoma patients consumed less meat than people without glaucoma. These are hints, not proof, that a higher protein diet may coincide with better eye health. Importantly, these studies used food questionnaires and could not prove cause and effect. It's possible that other lifestyle factors correlate with meat eating. Other studies have looked at overall dietary patterns that include protein sources. Notably, a combined Mediterranean DASH diet score enriched in fish, sometimes called the MIND diet, was found in a U.S. cohort to associate with lower glaucoma risk. Specifically, each 10% better adherence to the MIND diet, which counts fish twice a week, nuts, berries, vegetables, was linked to about a 20% reduction in new glaucoma cases. This suggests that diets rich in healthy proteins, like fish and plant foods, might be protective. In contrast, diets defined mainly by high animal protein with unknown overall quality have mixed evidence. At least the limited data we have suggests more meat was not harmful and perhaps helpful. Mediterranean and MIND dietary patterns. The Mediterranean diet, high in fruits, vegetables, whole grains, legumes, fish, and olive oil, is often examined for eye health. Evidence on glaucoma is still sparse. A recent comprehensive review covering many eye diseases noted that most studies find the Mediterranean diet clearly helps diseases like age-related macular degeneration and diabetic retinopathy, but the evidence for glaucoma is inconclusive. For glaucoma specifically, only a few studies exist. In Spain, one large prospective study found no significant link between a person's Mediterranean diet score and glaucoma. However, that study did find that a broader healthy lifestyle score, Mediterranean diet plus exercise plus no smoking, was associated with about half the incidence of glaucoma. Similarly, a Dutch study, the Rotterdam study, found no effect of the Mediterranean diet score alone on glaucoma, but did see a 20% lower odds of open-angle glaucoma in people following the related MIND diet. In short, some data hint that Mediterranean-style eating, especially with the emphasis on fish, nuts, and veggies, might help protect against glaucoma, but results are mixed and more research is needed. Nerve damage and vision loss. Few studies have directly linked diet to the structural or functional damage in glaucoma, retinal nerve fiber loss, or visual field defects. A scoping review of diet and glaucoma noted that of 19 relevant studies, only 2 included measurements like retinal nerve fiber layer thickness from OCT imaging or visual field testing in their outcomes. Those studies tended to focus on dietary nutrients, like vitamins, rather than broad macronutrient patterns. Overall, there is very little data yet on how diet affects glaucoma progression measures, such as nerve layer thinning or vision loss. Most evidence is about disease incidence or risk, not about measures like visual field loss or nerve fiber changes. This remains an important gap. Future research could look at whether, for example, high-sugar diets are linked to faster visual field decline in patients, or whether high healthy fat diets safeguard the nerve fiber layer over time. How macronutrients might affect eye pressure. Epidemiology hints at links between diet and glaucoma, but what mechanisms could explain this? Here are a few plausible pathways. Osmotic effects of sugar. When blood sugar rises sharply, as after a high-carb meal, it increases the osmosis, water-drawing power of the blood. This can draw water into the eye's fluid. Classic studies in glaucoma patients have shown that acute spikes in blood glucose are closely linked to increases in IOP. For example, one investigation monitored glaucoma patients during a glucose tolerance test and found that the eye pressure changes paralleled the blood sugar and blood osmolality rises. In practical terms, very high blood sugar makes the aqueous humor, the fluid inside the eye, more concentrated, pulling in water and transiently raising IOP. Chronic hyperglycemia in diabetes may likewise increase the eye's internal osmotic gradient, the difference in solute concentration between inside and outside the eye. In fact, a recent review highlights that diabetics tend to have higher IOP partly due to this osmotic effect of glucose. Thus, diets that produce large blood sugar swings, lots of refined carbs or sugars with little fiber, could acutely raise eye pressure. Conversely, diets that keep blood sugar steadier, low glycemic index foods, or medication for diabetes may help keep IOP lower. Insulin and eye fluid production. Along with raising blood sugar, eating carbohydrates triggers insulin release. Insulin itself can affect fluid and nutrient transport in the eye. The ciliary body, which produces the aqueous fluid, and the blood-ocular barriers have insulin receptors. Animal studies show that after meals, insulin levels in the bloodstream rise sharply, while insulin levels in the eye fluid rise more slowly and to a much lower peak. In a classic rabbit study, after feeding plasma insulin tripled and aqueous insulin roughly doubled, and the increase in eye fluid glucose closely tracked the insulin peak. This means that post-meal insulin exposure may influence how much glucose and water enter the eye. For instance, one human study gave insulin infusions to diabetics and measured intraocular fluid flow. They found that diabetes patients had lower baseline aqueous flow and that insulin levels influenced flow rates, although the exact details are complex. The bottom line is that systemic insulin dynamics can affect eye fluid dynamics. High insulin after a meal might change sodium and glucose transport in the eye, altering aqueous humor production or drainage. If diets, like very low-carb diets, change how much insulin is released, they could theoretically influence eye pressure through these mechanisms. More research is needed on exactly how insulin signaling in ocular tissues impacts IOP. Lipids and the trabecular meshwork. The trabecular meshwork, TM, is the spongy drainage tissue through which aqueous fluid leaves the eye. Its function largely determines IOP. Importantly, TM cells respond to various lipid signals. Modern lipidomic studies show that different fats are present in the aqueous humor and TM cells. These fats, phospholipids, sphingolipids, etc., affect the stiffness and contractility of the meshwork. A recent review notes that lipids in the eye fluid can change the biomechanical properties of the TM and thus alter fluid outflow. For example, prostaglandins, a type of lipid-derived molecule, are used as eye drops to lower IOP by relaxing the outflow pathways. Thus, the balance of dietary fats could influence which lipid signals the eye produces internally. Indeed, analyses of glaucoma patients versus controls reveal different lipid profiles. For instance, one study found that higher dietary intake of the omega-3 fats EPA and DHA, commonly from fish oil, was associated with a lower risk of glaucoma. On the other hand, diets very high in certain polyunsaturated fats, especially if rich in omega-6, were sometimes linked to higher IOP or glaucoma risk. Lab experiments also show that blocking lipid synthesis in TM cells, called de novo lipogenesis, can lower IOP in animal models. In summary, dietary fats may influence glaucoma via their effects on the TM and on chemical messengers, like prostaglandins, that control fluid outflow. A shift towards anti-inflammatory omega-3 fats, as in a Mediterranean diet, could help keep the drainage system pliable, whereas excess unhealthy fats might stiffen the TM or alter outflow negative. Gaps in the evidence and future directions. Overall, the evidence to date is intriguing but far from definitive. A recent review of diet and glaucoma studies found that most of the data are observational. 95% of studies are either cross-sectional or prospective cohorts. Only a handful of trials exist. For example, one analysis of a women's low-fat diet trial, and only 2 studies actually measured nutrient biomarkers alongside diet. In addition, diet has mostly been self-reported by questionnaires, which can have errors. Outcomes have typically been diagnosis of glaucoma, rather than direct measurements like nerve fiber thickness or field loss. Key gaps include lack of longitudinal interventional data. We need more prospective trials or long-term follow-ups to test if changing diet can alter eye pressure or nerve loss. For example, no randomized trial so far has assigned people to a Mediterranean versus control diet and measured IOP or visual field changes over time. Limited outcomes. Very few studies have combined diet data with clinical measures like OCT retinal nerve fiber thickness or standard visual field tests. Future research should include these objective glaucoma metrics to see if diet affects progression, not just diagnosis. Heterogeneous diet assessment. Different studies use different food questionnaires or nutrient scores, e.g., one study's Mediterranean score may not match another's. Standardizing dietary assessment, for instance, using validated food frequency questionnaires or food diaries, plus biomarkers, blood lipids, glucose, etc., would improve comparability. IOP phenotyping. It would help to measure IOP more precisely. For example, instead of a single office measurement, future studies could use 24-hour IOP monitoring or multiple measurements to capture fluctuations. Accounting for corneal thickness, pachymetry, and ocular perfusion could also refine risk assessments. Specific macronutrient measures. Rather than broad categories, researchers could quantify exact macronutrient percentages of diet or glycemic load and relate those to IOP changes. Measuring post-meal versus fasting IOP could test the glucose-insulin hypotheses directly. Addressing these gaps will require collaboration between ophthalmology and nutrition researchers. Large cohorts should incorporate diet surveys and regularly scheduled eye exams with IOP, OCT, and field testing. Randomized feeding trials, even short-term, like a few weeks on different diets, could examine acute effects on IOP and eye fluid composition. Finally, mechanistic studies in labs or animals can test how altering glucose, insulin, or specific fats changes the trabecular meshwork at the cellular level. Conclusion. In summary, there is growing evidence that what we eat may influence eye pressure and glaucoma. High blood sugar appears to raise intraocular pressure through osmotic effects, and diets that minimize sugar surges, e.g. low-carb or low-glycemic diets, could theoretically help keep pressure lower. Dietary fats also play a role. Some studies find that healthy omega-3 fats from fish or plants are associated with lower glaucoma risk, whereas other fat patterns or extreme low-fat diets do not appear protective. Diets rich in vegetables, fruits, fish, and lean proteins, as in Mediterranean or Main diets, have shown some promise for reducing glaucoma risk. However, the data are far from certain. Most studies so far are observational and use self-reported questionnaires, so they can only show associations, not prove cause and effect. There are many unknowns. For instance, whether any diet change can slow the actual progression of visual loss. Nonetheless, these findings are exciting because they hint that lifestyle measures, diet changes, might one day complement glaucoma treatments. For patients and clinicians, focusing on a balanced diet with plenty of vegetables, healthy fats like fish or olive oil, and moderate protein while avoiding high sugar loads is prudent for general health and may also benefit the eyes. Going forward, eye doctors and researchers should design studies that carefully track diet and eye outcomes together. This includes using standardized nutrition surveys or recalls, collecting blood markers like glucose spikes or fatty acid levels, and performing regular eye pressure and imaging tests. By bridging nutrition science and ophthalmology, we can better understand if and how macronutrient patterns truly affect the health of the optic nerve and help prevent vision loss from glaucoma. All links to sources are available in the text version of this article. You can find the full article at visualfieldtest.com. Thanks for listening. To check your visual field, click the link at the bottom of this article or visit visualfieldtest.com.